All right, hi everyone. We're live here at the Agler 2017 poster session. Keep tuning in because we're going to have not only Jess here in just a moment, but we'll also have a couple other posters coming up. Ellen and Ariel will be on shortly, so keep tuning in. Any questions you have for any of our poster presenters, definitely send those in. If you don't get to watch it while it's live and you have questions, definitely feel free to tweet them at their respective handles. So with that, I'm going to turn it over to Jess Sherman-Collier. I keep wanting to call you Jess Sherman.
You can call me Jess Sherman, that's okay.
Okay. When you get married, someone says hi. So, but yeah, so we're going to turn it over to her, let her talk about all of her amazing Great Lakes mussel work.
Hi everyone, thanks for joining us at Agler 2017. I'm Jessica Sherman-Collier sometimes, and I will be talking about my mussel work in the Maumee River. So for those of you who may not know about my research, I work mostly with lake sturgeon, and I started a project for lake sturgeon habitat suitability modeling to reintroduce them into the Maumee River. So once we created this model, we wanted to know, could that model then be used for any other species in the Maumee River? So we decided to test it out with the native freshwater mussels, which we call uniatids. And you can see an idea of our uniatids here. This is just a portion of some of the assemblages that we found. So for our initial model, we were using the characteristics of substrate, water depth, and water velocity, and we categorized these out as either poor, moderate, or good based on uniatid preferences from the literature. And we created this overall habitat suitability model for uniatids that was depicting areas that we thought would be good, moderate, or poor. And so our idea was that in these good areas, that's where we'd find our highest abundance and highest richness of uniatids. In the moderate areas, that's where we'd find moderate abundance and richness. And in the poor areas, that's where we'd find our low abundance and richness. So what we did then was we randomly selected areas equally distributed among good, moderate, and poor. And there might be hard to see, but there are these little dots all along the river. And when we went out and did habitat surveys, they were 10 by 10 meter quadrats of the river. We searched them with tactile, so hand searches and clam rakes, and we did this till we exhausted the area. So it wasn't necessarily a timed search, it was until the last mussel was found and 5 minutes after that. So we were trying to exhaust to make sure that there weren't any uniatids left behind. And then the mussels that we found, we keyed them out to their species and their size, and then we had we looked at the richness and abundance that was found at each site, and to determine if we found the most mussels in our good areas and the least in our poor, we did a Poisson regression model. So it's a generalized linear model that was just analyzing the number of mussels that we found and the number of species we found in each of the categories, good, moderate, and poor. And what our model found was that, yes, in our good habitats we had our highest abundance, so our highest number of mussels, and in the poor habitats we had the lowest. But as far as species richness, our model wasn't able to delineate out habitats that had either good or moderate or poor species richness. It was kind of neutral across the board. So our model works in terms of predicting where we can find mussels. The good part about this model is for scientists or even advocacy groups that have a home river system or stream system, they don't even know where to begin. So in the Maumee River, the section that we were surveying was 56 river kilometers. That's a pretty long distance. And if you don't know where to begin looking for mussels, you don't know what your assemblage and community looks like in your whole river system, how do you begin to do those surveys to find out what is there? And so this model will give you an idea of areas where you can start looking. So for the model that we predicted those good habitats in these areas, that's a good starting point to go out and survey. And we wanted to create a tool for people that they'd be able to use, that they can then go out and say, okay, I have this system, we don't know what's in it, and we don't know where to start. And you can use this tool as a starting point. And so that was the main goal for it. It transferred from sturgeon to uniatids. It's not a perfect model, but it's a great starting point.
Awesome. So I guess where else in the Great Lakes, can you find them, like in rivers, lakes. Where area that you always are finding these? Like, where's the best places to find them besides the Maumee River?
Yeah, so you can find Uniatids in rivers and out in the lake proper. There's different species. So our species of mussels in the Maumee River, fast flowing. Some areas are kind of rocky, and these species are more adapted for those river systems. So they have thicker shells. Some of them, if you can see it on the photos, they have these little nodules on their shells that help anchor them into the sediments, so they don't get dislodged by the current. Out in the lake proper, where we don't necessarily see the same types of currents and fast water velocities in the river system, those species of mussels are usually a thinner shelled species. And so historically, you could find mussels all across the Great Lakes, except for the deepest parts of the lakes, but most of the littoral or edge zones, and then throughout nearly all of our river systems. Now, because we have altered our habitats with dams and we've changed the sediment inputs, there's a lot of sedimentation and eutrophication into our rivers and the lakes. We have invasive species that will eat them or foul and kill them or outcompete them. So our populations of Uniatids have been imperiled across the Great Lakes. Freshwater mussels are the most imperiled group of organisms in North America, and in the Maumee River, 18 of our species are listed as imperiled.
Wow, that's quite a lot.
That is a lot.
Well, thank you, Jess.
Yeah.
If anybody else has questions for Jess afterwards, you can tweet her at aqua_amare.
Yeah.
So, and just look her up on Twitter, and she'll definitely answer any of your questions when it comes to mussels, sturgeon also.
And sturgeon as well, yes.
Yeah. And so for everyone else out there, stick around. We'll be putting up the links for the next couple posters here, basically as soon as I can go find them. I know they're somewhere in this mass of posters back behind me. So till then, thank you for tuning in, and get those questions in because we love that engagement. Thank you.